Hello, today I will be presenting a review article which summarizes Dr. Jaubri's contributions to the field of immunology, specifically her work in celiac disease. As we know, Dr. Bana Jaubri is a doctor in pediatric gastroenterology. Her research focuses on autoimmune disorders, irritable bowel syndrome, gut health, and celiac disease. She has been with the University of Chicago Department of Medicine and Celiac Disease Center since 1999. In 2009, she received the William K. Warren Jr. Prize for excellence in celiac disease research. The picture in the top right corner shows Dr. Jaubri and a student looking at a figure from the paper that I presented in the last video. Now let's take a look at her contributions thus far. A review of Dr. Jaubri's contributions in her field show her impact on understanding the molecular pathways involved in celiac disease. By identifying the key effector cells mediating celiac disease, Dr. Jaubri was able to create the first animal model for celiac disease. This model allows for more research and treatment options for patients. Dr. Jaubri was also able to determine that the chronic inflammation triggered by active celiac disease changes the T-cell receptor intraepithelial lymphocytes that reside in the tissues of the small intestine. Lastly, Dr. Jaubri found evidence that the BNZ2 enzyme blocked IL-15 and IL-21 transcriptional alterations. Remember, IL-15 and IL-21 are key effector cells mediating the villus atrophy associated with chronic inflammation. According to the University of Chicago, her current research is focused on discovering markers that can identify an abnormal reaction to gluten before antibodies are present. The role of gut microbiota in the pathogenesis of celiac disease and potential therapeutic implications, published in July of 2020, provides an overview of previously published data on how potential mechanisms cause celiac disease through the gut microbiota. The figure on the right is an interesting flowchart showing their method of literature selection. Dr. Jaubri's work was referenced throughout the review article often, specifically her work in determining how gluten proteins disturb the host epithelial functions. The modulating HLA-DQ8 and DQ2 haplotypes, microbial composition differences in fecal and duodenal samples from patients with active celiac disease compared to healthy controls. This is where she found the most abundant bacterial phylum in active patients. She also found a trend between a reovirus infection and the pathogenesis of celiac disease. In Figure 2 of the review article, the molecular pathway of celiac disease is shown in its entirety. This figure highlights the important contributions of Dr. Jaubri. Near the center of the graphic, we can see the relationship that IL-15, TNF, and IL-21, as well as the interferons, have with each other and their consequences on the immune system. The top of the graphic illustrates the microbiota type and the potential impact on the pathogenesis of celiac disease, analyzed by the article. The rightmost section of the graphic specifically highlights the portions of the pathway determined by the work of Dr. Bana Jaubri. The key effector cells of epithelial cell damage addressed in Dr. Jaubri's papers are shown in the top portion, as well as the genetically susceptible haplotypes HLA-DQ8 and DQ2. Another cited work in T cells is highlighted in the bottom portion of the graph. Dr. Jaubri has published important work, specifically in the field of celiac disease research, that has fundamentally laid the groundwork for future research in therapeutic treatment and causes of the disease. If you have any questions, feel free to leave them below, and I will get to them as soon as I can. Thank you.